Welcome back to Can You Hear Me, a podcast about hearing, hearing loss, and everything in between. I'm joined today by Dr. Julie Arenberg, Associate Professor of Otolaryngology at Harvard Medical School and Director of Auditory Research and Education at Mass and Eye and Ear. Dr. Arenberg, thank you for taking the time to be on my podcast today.
Thanks for having me.
Of course, and I'm glad to have you on too. So, my first question for you is about your background. Could you tell me more about how you first became interested in working in hearing and cochlear implants?
Sure. I sort of grew up in it, so to speak. My father is a retired neurotologist, and so I grew up hearing about audiology and hearing research. But then when I was in college at Boston University, I had this class that really sparked my interest in actually the hearing and the brain. It was called physiological psychology. And from then on, I just wanted to understand how the brain was working and influencing our behavior. And it led back to hearing related research in graduate school.
Yeah, that's really cool. So, you really have been in this field to some degree, really since you were a child. So, that's really cool because I see a lot of other people who were introduced, I'd say, in college or later, but it's kind of cool that you were exposed to this from a really young age and that you kept through and you liked it. So, just to dive straight into some questions about your research, my first question for you is about your lab, the ear lab, the Emerging Auditory Research Lab. And there I saw one of your ongoing projects is titled Development and Assessment of Listener-Tailored Programming for Cochlear Implant Listeners. Could you tell me some more about this study? I also saw that it focuses specifically on patients with advanced bionic cochlear implants. And just for my listeners who don't know, Advanced Bionics is one of the three major companies that produces cochlear implants, and they have around a 20% market share.
Yeah, so it's interesting. I'm probably one of the few people who has continued their work from their PhD all the way through their career. But I started out studying in animal models, different ways to activate the auditory system. We were looking at cochlear implant stimulation and how could we get this electrical signal to activate the brain in a similar way to how the brain's activated by acoustic sound in normal hearing animals. And so we came up with a way that focused the size of the electrical field, and that better mimicked the brain responses to pure tones, which are sort of the purest sound that we use. And from there, after I finished my PhD, I realized I was much more of a people person than an animal person. And so I actually got my clinical degree in audiology. And I also, at that very time in about 2001, Advanced Bionics had come out with some research tools that allowed me to test for the first time how these signals were perceived by patients with cochlear implants. And so that started a long collaboration with Advanced Bionics. Their cochlear implant, until very recently, was the only one on the market that could stimulate focused electrical fields in the way that I was studying. So their device has a current source for each of the electrode contacts in the cochlear implant, which has anywhere from 12 to 22 in the implants on the market. And so this allows people to steer the current between electrodes or focus the current within the inner ear. So we studied that, and we then developed methods to use that in a speech processing strategy. with the idea being that if we could activate the brain more similarly to how it responds to normal sound, then people would understand speech better with our strategy. And so that's sort of the foundation of that study. And now we're working to implement the study in people across the lifespan, so in children and adults, and to determine how to program it so that it restores the most hearing we can for people with hearing loss and cochlear implants.
Yeah, that's very exciting, and it's great that you're able to continue your PhD work because I've seen a lot of professors in the field who their PhD is in one thing, but their current research has strayed so far away from that. So it's really nice that part of your work is still rooted in when you got your PhD. I think that's awesome. I just had a follow-up question on one of the things you said. You mentioned using animal models. Could you tell me more about how those models were important to your research or what kind of things you used to do with them? And also, when you mentioned, uh, more focused electrode stimulation, was that more of a hardware or like a software kind of thing?
Yes. So the animal models we worked on, and actually still I'm working on them again now in some other studies. We use the guinea pig model because their cochlea is easily accessible and their hearing range isn't so different from humans, the sound frequencies that they can hear. And animal models are really valuable for developing novel ways to stimulate the auditory system. And also, because you can measure brain activity at different in different parts of the auditory pathway, it allows you to really understand what kind of signals you're using to drive the system, and you can compare them directly then to what we know about normal processing in the auditory nervous system. And so it's really valuable that way. I don't remember your other question.
Oh, well, the other question, well, I think you answered the animal model question. It was more the electrode focus. Was that
Uh, is it a
the
hardware or
hardware
software?
software? Yes.
Yes. Um, it's a little bit of both. So, like I mentioned, the Advanced Bionics device has a current source for every electrode contact. And so that's the hardware part. But then we can control in software where the active electrode current flows and how we return the current path. So in when you make a broad electrical field, which is mostly what's done clinically, you're stimulating one electrode inside the cochlea and a return electrode outside of the cochlea, and that makes this broad electrical field. When we focus the electrical field, we can return the current to electrodes within the cochlea, and that keeps it more contained and activates a more restricted population of neurons.
I see, I see. And is this something to do with the monopolar versus tripolar stimulation techniques?
Yes, so the broad one is called monopolar, and the more focused one that we've been studying is tripolar.
I see, I see. So just could you sum up why tripolar electrode arrangements aren't clinically popular? Because I've read some things in the past and I've seen that tripolar tends to, as you said, it's able to give a more focused electrical signal and stimulate a more focused population of neurons. But I'm really interested in knowing why there isn't so much more of a push to get these clinically translated. Because to me, at least my first impression is that if you're stimulating a more select group of neurons, the voltage would be less, so you would actually be using less power. But I've also seen other studies that have shown that you're actually using more power and it takes more from the battery life of the implant. So I'm really curious what your explanation for this is.
Yeah, so we've been studying it for a long time, and there's a couple of reasons I think that it hasn't translated yet. One of them is when you use a true tripolar stimulation, um, it does require more power because for the brain to process sound, you need to activate a certain number of neurons. So if you keep the field really small, um, you won't activate enough neurons for the listener to perceive sound, and it won't get loud very quickly. And so then it does require more power. Um, Patients who we've made these programs with also reported that the sound quality wasn't as full or rich as it is with monopolar. And we saw this also in our animal models, that the neural activation didn't spread as much as it does with normal acoustic sound. And so we then came up with a dynamic tripolar strategy so that we could increase the spread to be more natural, but still maintain a focus when the brain is normally getting a focused input. So for soft sounds, the stimulation is more restricted than it is for loud, and then you get a more natural activation of the auditory system. And when we do this, it also requires a lower amount of power. And so this is what we're hoping we can translate into the clinic.
Yeah, and I'm really excited to see maybe in the next decade or possibly even further how this kind of technology evolves. And really, it's interesting for me as I read about the history of cochlear implants to see that the first ones were single channel. They only had one electrode that was even in the cochlea, stimulating all these hair cells. So to see for a while how everyone was just trying to get more focused activation, more electrodes. But I guess at a certain point, it's become less of that and more of how can we optimize the software to just give better speech perception without actually having more electrodes, because I guess that doesn't really help as directly as it used to in the past, as you were saying with the tripolar versus monopolar, if I'm interpreting what you said correctly.
Yeah, more or less. Although one important thing is that the cochlear implant doesn't activate the hair cells per se, because people who have severe or profound hearing loss, usually their hair cells are degenerated. And so the implant bypasses those degenerated cells and activates the auditory nerve directly.
Yes, sorry. Yes, that I meant the neurons. I'm not exactly sure what they are called, but the neurons of the auditory nerve in that area.
Yeah.
But yes, thank you for that. My next question for you is, how do you choose what projects you work on? Is there a specific theme in otology or audiology or something that inspires your work?
Hmm. Yeah, that's a problem I have, is that I'm so inspired by so many different projects that I am involved in a lot of um, different work. I usually I feel like when we do research, we end up answering some of the questions we are trying to answer with various experiments, but usually we open up more questions that need to be answered. And so I often follow that vein to the next most logical questions. I'm always thinking of what's going to translate because of my clinical training. I feel like I'm always thinking, well, what would be useful clinically? How can we make this a more viable strategy or something? How do we make it accessible to the audiologist to program for patients? Um, but here, since I joined Mass Eye and Ear a few years ago, I've become more involved in things related to ototoxic monitoring and developing new audiology protocols and evidence-based practice. And I end up being passionate about a lot of different areas related to hearing because I think it's just so important, and it's just there's a fascinating um, amount of work to still be done. So
Yeah, I was just, oh, sorry to interrupt, but yeah, I completely agree with you. I mean, it's so interesting, and even in my limited research knowledge and experience, I'm seeing so many different aspects of hearing loss research come into play. I mean, there's genetics where people are looking at, some types of genetic hearing loss or even the gene therapy stuff. And then there's your kind of research, which is looking more at clinical translation and the audiology and cochlear implant aspect of it. But there's also so much research being done to look at the underlying neural pathways from the central auditory pathway and just even higher stuff in auditory cortex. So really there's so much to investigate, and that's really why I wanted to ask you that question because as you said, really research is better at bringing up more questions than answering the ones we have. And I see that too because in the projects I work on, I'm always asking my mentors, like, can we investigate this or can we investigate that? And they always tell me, that's a separate project. Let's focus on this one and then we can get to that. So I really do agree with you when you say that.
Yeah, it's hard to stay focused on one project when there are so many interesting questions to ask.
Uh, yes, really, that's true. My next and final question for you is how would you say your lab's work fits in with the overall aim of the Eaton Peabody Laboratories? And just for my audience, the Eaton Peabody Laboratories are at Mass Eye and Ear and are among the largest and longest standing auditory research centers.
Yeah, I feel very lucky to be part of the Eaton Peabody Labs here. It's an amazing group of investigators and incredible trainees. Um, I feel like the work at Eaton Peabody Lab really spans the whole range that you just talked about. There are people looking at genetics, there are people looking at gene therapies, there are people looking at regenerating neurons and hair cells. There are lots of people using animal models to study the auditory nerve and the inner hair cell and the synapse between them and the brainstem and the auditory cortex and then there's a few of us who are doing human perception and neurophysiology with like electroencephalography, with EEG and other measures. And so I think we really span the whole gamut of what's possible in auditory research. And it's just a really, it's an incredible place to work and to collaborate with so many brilliant people. It's a great opportunity.
Yeah, and I can really tell when I see all of the works that Eaton Peabody puts out. I mean, it's really amazing. I know I keep bringing this up, but how in hearing loss, which is in itself a kind of one concept, there's just so many different research fields within that are being attacked by so many different investigators. And this is even just in hearing, which is of all the medical specialties, I guess one of the smaller ones. So really it's so interesting just how many aspects and layers there are to all of this. So just to close off today, are there any last remarks you had?
No, I mean, I just want to thank you for putting this out there and for letting people know what important work is being done here to try and improve hearing for people.
And I appreciate that. So Dr. Arenberg, thank you so much for your time today. I really enjoyed our discussion. I know I asked a lot of follow-ups, but really I do find all of this work extremely interesting.
Thank you.